thank you. First of all, thank you to the organizers for inviting me here. So is that good enough? You can hear me? Okay. Yeah, thank you again for inviting me here. It's a great pleasure for me to present non-clinical development of CAR-T therapy products. So I will speak very slowly to give the translators the opportunity to adequately translate. Okay, the agenda is as shown here. We will have some very general remarks on how CAR therapy works, then that it makes a difference in certain diseases, and then we will rapidly evolve into the risk associated with CAR-T therapies, what they are, how they can be tackled, and what the non-clinical risk assessment with regard to this could be. And in the backup, I don't think that we will go through that. I have also some regulatory environment information from the EMA and the FDA. Okay, so it was initially an idea by Selig Escher, who worked at the Weizmann Institute and the National Cancer Institute, together with Steve Rosenberg, to arm T cells in a way that they could fight cancer cells. And they did that following Escher's idea by introducing CARs, chimeric antigen receptors, into T cells by genetic modification. Okay, that's this one here. And the first attempt in the clinic was not very successful because it turned out that the CAR was not efficacious enough and the cells were not responding enough when targeting the tumor cells. So they had to introduce secondary co-stimulatory domains into the transgene, which are expressed then in the T cell, and that was a success at the end. What I would like to draw your attention on is that we are talking today only about chimeric antigen receptors, and I say shortly CAR, to differentiate from T-cell engineered receptors, which are dependent on MHC presentation by cancer-relevant peptides. So we are not talking about that. So how does CAR therapy work? Shortly, blood is drawn from a patient, undergoes apheresis, and then you collect the T cells and isolate the T cells, which are transduced with a vector who carries the desired transgene of a CAR directed against CD19 or BCMA, for example, B-cell maturation antigen. And then the cells are usually kept in culture to give them the opportunity to proliferate. And after that is done, the cells are packaged, sent back to the hospital, and reinfused into the patient with the aim that the cells are fighting the cancer in the patient. So this shows how busy the whole field of, in the immuno-oncology landscape, how busy the field of adoptive cellular therapy is. What we are talking about today is the autologous CAR, but you also have autologous TCR engineering, and you have allogeneic approaches for CAR T cells and also TCR engineered cells to make them available from the shelf and not taking them from the patient, the cells. And also other cells are mentioned here, gamma delta T cells, natural killer CAR cells, and so on. We are focusing on that area here for today. This list shows you the approvals by EMA and FDA. Okay, I have this sign here on my screen, and I was wondering if that's also on the big screen, but it's not the case. So this shows the chronology of the approvals of CAR-T therapies by EMA and FDA. So first there was Kymriah or tisagenlecleucel, a CAR against CD19, approved in 2017 by the FDA, and closely followed by the Yescarta card in October 2017, and then later on in the EMA in 2018 also both were approved. And you see also that they were followed by several other CARs, which are approved in Europe and the United States, and also are going to be approved in other areas of the world. And you see that we are mostly dealing here with third line, fourth line, sometimes second line treatment, but the field now is really rapidly moving into first line treatment of severe leukemia and lymphoma, for example. So a lot is happening in this field. It's very busy, and a lot of innovative activity is going on there. One example, of course, since I'm from Novartis, I'm showing Kymriah results with remarkable efficacy, manageable safety in the clinic, and durable benefit. We have treated, this is already an old number, we have treated over 9,000 patients with Kymriah. And especially if you look in ALL cancers in children and young adults, you see that we have outstanding overall survival here, response rate, complete remission over 80%. It's also true in follicular lymphoma. It's a bit less efficacious in DLBCL, diffuse large B-cell lymphoma, because of the patient conditions which are different to those of ALL and follicular lymphoma. But nevertheless, this was the first time that in oncology something like this could be presented, and also not only from Novartis, but also from in the meantime, from other companies. Now, with being so successful in the oncology field, the researchers are moving into a non-onc indication in severe refractory systemic lupus erythematosus or lupus nephritis. And what I show here are results from a clinical group by Georg Schett from the University Hospital in Erlangen in Germany. And you see here, they used a CAR directed against CD19. That means the aim was to deplete the B cells in the patient and also the autoreactive B cells by that. And you see here that, without going into the detail of all the endpoints of the measurements here, but it's summarized in the so-called SLEDAI score. You see that with the time after the CAR infusion, all these markers go down to normal and are no longer visible here. And you see also that after 20 months, patients are still in remission here. Okay, it's a few number of patients. I think this was from last year. It may be a bit more this time. But you see that these are outstanding results, and the patients are doing very well, although they were refractory to standard of care treatment before. Okay, let us come now to the potential risks which are associated with CAR T therapy. Of course, we have to consider on and off target toxicities by the CAR. I will come back later to that. Target selection and validation is very important in that field here. Then we have the well-known effect of cytokine release macrophage activation syndrome in patients. This was not discovered in non-clinical studies, and the reason for that I'm coming later to. It's considered a pharmacodynamic effect of the CAR T cells. And in some cases, the more tumor cells are in the patient, the stronger the CAR T cells get activated, and the stronger also in some patients the cytokine release syndrome can be. And so that's why it's a pharmacodynamic, an exaggerated pharmacodynamic effect. Tumor lysis syndrome is also a known effect from standard of care treatment. If you very rapidly destroy cancer cells, then their content is released into the organism and will do these adverse effects there. Neurological effects are in the context of cytokine release syndrome. They appear a little bit later, were also not discovered in the non-clinical models which are available. Cytopenia is related to lymphodepletion of the patients because to enforce the engraftment of the CAR T cells, the patients are lymphodepleted by fludarabine and cyclophosphamide. And it's also sometimes related to cytokine release syndrome. Very importantly, and we will have also some slides on that later on, insertional mutagenesis, and in principle, an oligo or monoclonality can lead to an insertional oncogenesis. We will come back later to that. Of course, the CARs targeting B cells or leukemia cells, they at the same time target also the healthy B cells. You will have B cell depletion, which is also considered a PD effect and can be monitored in the clinic and can also be treated with immunoglobulin substitution. Then we have a potential impact on the fetus if a female patient gets pregnant. There is an effect known as maternal microchimerism. You have fetal and maternal microchimerism, and it depends on the direction where lymphocytes can cross the placenta. This is a well-known effect and has also been brought into connection with the development of certain autoimmune diseases by childbearing women and also by newborn children. There are no standard animal models available for this to investigate that, and there is some research ongoing on that, but at the moment we cannot say anything to this. That's why strict contraception in the clinic is required for CAR T therapy, especially in the first 1 or 2 years of the therapy, where the counts of the cells may be pretty high. Okay, so why is target selection so important for safety? So here's one example which was published or which was described in a review. I recommend to read that review by Ulrich et al. To everyone. It's very interesting. It's summarizing several cases of clinical safety and efficacy effects by CARs and T-cell engineered cells. And it's a good collection of what can happen. And it shows also that you really have to take care on the target selection and look where it is expressed apart from your tumor tissue. And in that case, the CAR was directed against carbonic anhydrase and in a renal cell carcinoma trial. And the tissue distribution of the target was shown to exist in the liver, the small intestine, and the gastric mucosa. And in this clinical trial, it didn't come to any efficacy readout because the treatment had to stop very rapidly, or patients had to be treated with steroids, which is turning down the CAR activity also. And but what they found instead was liver toxicity, and immunohistochemistry of the liver showed also that the CARs were present there and doing their job there. So it's this is one example of on-target off-tumor toxicity. On the other hand, if we are dealing with off-target toxicity and how to address that preclinically, there is since a few years a so-called plasma membrane protein microarray available by another company called Retrogenics, but there are also other companies providing that in the meantime. They have up to now, I think, over 6,000 membrane protein in a HEC293 based microarray where you can test your chimeric antigen receptors as a single molecule and can identify potential cross-reactivities. And this is accepted also by FDA and EMA. It's also accepted by antibodies, therapeutic antibodies in the oncology field, instead of doing a GLP tissue cross-reactivity study on human tissues. Okay, so oncogenicity risks of the current retroviral gene therapies. You all may have heard about the cases which happened after gamma retroviral therapy. That's important because we are talking, Kymriah, for example, is a lentiviral product and several others also, whereas Yescarta is a retroviral product, I think. And so there had been cases in the past, the famous in the beginning of the 2000s, end of the 90s of the last century, the cases of the severe combined immunodeficiency boys. It was a disease, an inherited disease affecting boys only, they underwent a gene therapy where they corrected the gene with a vector in the autologous hematopoietic stem cells, re-injected them, and then those boys, after a few time, or some of them, developed a T-cell leukemia. How could that happen? Is because a hematopoietic stem cell is subject to delineation and complex genetic regeneration, and completely other genes than in an end-differentiated T-cell will be active here. And in that case, and because a retroviral vector was used, which has its own safety risks here bringing with it, the insertion was in the LMO2 site, LMO2 gene in the hematopoietic stem cells, which is responsible for the maturation of T cells. And that at the end led then to T-cell lymphoma in those boys. Okay, so how do we now avoid these cases, for example, of introducing that? First of all, it's not only the vector, it's also the target cell, because I said that T cells are more end-differentiated than hematopoietic stem cells, and they underwent epigenetic regulation also of genes. Most of the genes of the HSC are silent, and others are active now. And the other thing is that the vector itself was changed, also the lentiviral vector. It turned into a self-inactivating format in the long terminal repeat. Then it was the whole production of the vector was done in a way that all the genes which are necessary to produce a functional vector were placed on different plasmids, following the divide and conquer approach, so that you control that, what is happening later on. And this made sure that at the vector at the end, only those genes are present which are necessary for reverse transcription and for integration in the genome, but not for packaging and producing new vectors. Okay, I think I have said everything for this case here. Yes. Okay, insertion site analysis is one method to investigate where exactly a vector can insert into the genome. When we were developing Kymriah, we were not asked by the FDA to do that. And we were also not asked by most of the European health authorities to do that, but there was one health authority in Europe who wanted to see that insertion site profile of Kymriah, and that's why we did that. And then we thought, okay, how can we get the most benefit out of these studies? And then we thought, okay, doing that only in a laboratory in the United States, we thought it would be good for several reasons to do that also in a lab in Europe with exactly the same samples. And that's what we did. We used 6 ALL samples, 6 DLBCL samples, and 2 samples from healthy volunteers, where we made final Kymriah products from, so ready for infusion in a patient. And this underwent then the lentiviral insertion site analysis. So I don't go into the details here, it's too complicated. And I'm also not a deep expert on this. But what I can tell you is that if you look on this stacked bar graph here, is if an insertion occurs over a certain threshold, then you will have more colored areas here. If you see gray, then most of the insertions are randomly distributed, and the abundance of that is below a certain threshold. Here, the positive controls, you see that there were preferred sites of integration, and that's why here you have in the positive control the red color here over the whole area here. So that's in short what this analysis is doing, and the outcome was from both labs with slight differences, but with the same overall conclusion that there is no risk which could be drawn from this lentiviral insertion site analysis. And there were also no substantial hotspots of integration identified in this study. Okay, then CAR-T clonality. That means the outgrowth of a specific clone in a patient of a CAR T cell. And we all know that clonality is a prerequisite of oncogenicity or malignancy. And we have seen that not with our cases with Kymriah, but where there were 2 cases out in the literature. And to my knowledge, these are the only cases in all these thousands of patients which appeared of CAR T clonality, but not with malignancy. That's very important. And how did that work? The cases were that in one case there was a CLL patient, chronic lymphocytic leukemia patient, who received the CAR therapy, and after a while the clone dominated his blood, the cancer was eradicated, the clone went down again, and the patient was in remission. And there was no sign of a T-cell lymphoma, even after years. So what happened here? It happened that the card inserted in the so-called TET2 gene, which is active in the epigenetic regulation of T-cell proliferation, and destroyed it. Usually the other allele takes over the function, but in this patient, he had an inherited mutation in exactly this gene, and then it was a complete knockout of the gene, and that led to this effect. However, it was beneficial for the patient at the end. Another case was a CD22 card by another group, and there was a disruption of the CBL gene observed, and it was a similar effect. We had a clonality, and after that the cancer was eradicated, the clonality disappeared again without developing a T-cell lymphoma or a malignancy. And if you have more interest here, I recommend to read the paper from Bushman of 2020. You have some explanations of that. Okay, another point when it comes up to the risks connected to the vector is the potential risk of replication-competent retrovirus or lentivirus. And I initially said already that we use a 3 or 4 plasmid system where all the genes responsible for generating the vector are distributed over different plasmids. Here is a 3 plasmid system. But you see that in the producing cell, which are mostly HEC293 cells or modifications from them, they release at the end replication incompetent lentiviral particles. So these are not able to replicate themselves anymore. And there are purification steps also shunted behind the isolation of those particles, and there are also tests made, RCL tests, to make sure that there is no replication competent vector anymore in the vector batch product. And in addition, also I already mentioned that this vector architecture, the self-inactivating format of the long terminal repeat, the 4 plasmid system, and the vector manufacturing, stringent RCR testing of vector product batch, and the accumulated manufacturing and clinical experience with the vector species. And even if it has a different cargo or transgene, you can use that to ask for a waiver of RCL testing at health authorities. In principle, both FDA and EMA require those testing, but we have good reasons, and we have for Kymriah also, we don't do RCR testing for the clinical product, for the patient batches, but for the vector batches, they're just an important feature. Okay. So this is in the new guidance from the FDA laid down from 2020, and there's also a very interesting explaining publication by Cornetta et al. From this year. If you're interested, you can have a bit more also scientific information on that. Okay, CAR T therapy and potential adverse effects like cytokine release syndrome. are connected to the activation and proliferation of CAR T cells. And the neurotox syndrome, the ICANS, is somehow connected in most of the cases to the cytokine release syndrome. As you can see here, it appears later after the CRS. Just quickly explaining here, you see the lymphodepletion conditioning of the patient with fludarabine and cyclophosphamide. That's the infusion of the CAR T. Then it takes a bit of time until the cells are proliferating and fighting against the leukemia, for example. And then during the activation, CRS develops. And this is accompanied by a set of, or a cascade of cytokines which are expressed here. And you see here that IL-6, for example, is one of the central cytokines of the cytokine release syndrome, and it's also a target of a clinical management of cytokine release syndrome by using tocilizumab, which is an antibody against the IL-6 receptor, and you can control that by applying this antibody. Okay, macrophage activation syndrome sometimes is also in connection to cytokine release syndrome coming on that, appearing in patients. It's also a pharmacologically exaggerated effect here. Currently, there are no standardized animal models available to investigate these toxicities. And the main reason for the inadequacy of animal models is the missing crosstalk between the human gene therapy product and the animal reticuloendothelial system. So that's very important to consider. Nevertheless, there have been some models developed in the last, in the 2000s to support first-in-man trials with the first CAR Ts. There was an homologous model which used biopsy mice, I think. And they were established with a blood cancer, these mice, and then there was a mouse-specific CAR T approach was used here. So this is not the clinical material which goes into the patient. It's a specialized product for the mouse, and also I think the vector sometimes has to be adapted to the mouse system because it's not so easy to apply vectors for humans also to animals. So, okay, I said the downside is it's not the clinical material, it's an homologous model. On the other hand, you can use so-called farm-tox hybrid studies using a tumor xenograft, which is established in immunocompromised mice. These mice do not express or do not develop T cells, B cells, and natural killer cells, and that's why they mostly accept the xenograft tumor and also human CAR T cells. Here you can use a GMP manufactured product, but you don't have tissue crosstalk between the CAR T and the mouse, and also the target of CD19 in the case of Kymriah is not cannot be targeted in the mouse because it's the epitope is not there. And anyway, these mice don't have B cells, so they don't have CD19 at all. Yeah, okay, that's also the reason, the missing crosstalk, why you can't see CRS and ICANS. It's also difficult, the biodistribution in that case, because you have human cells in a mouse environment, and here graft versus host disease reactions are the limiting factor, because the CAR T cells after a while start to react against the mouse tissue. And that's why these studies are limited. So as a standard, over the thumb rule, 3 months, but up to 6 months is possible. And I have seen papers where they described even more than 6 months, but this may be very specific to the conditions of the card, of the tumor, and of the animals which were used. So, in fact, the pivotal non-clinical study used for first in man in the US as a homologous model was used for Yescarta. So that was accepted by FDA and later on also by EMA. And okay, this one. And this is the one which was used for Kymriah. This was done by Mike Malone at University of Pennsylvania and published in 2009, and that was before Novartis took over. Just to mention that this was only partially GLP-like, because usually you cannot put this kind of studies into GLP conditions because there are no routine toxicity studies. And usually you try to mitigate that lack by putting the histology and the clinical pathology laboratory results or measurements under GLP, which are then also done in respective contract research organizations. So we come now to a bit more complicated slide here. This describes the study by Mike Malone, and I would like to explain why these models are limited for the use of CAR-Ts in the non-clinical area. So what you see here is that a mouse was established with a primary human ALL leukemia, and after a while, T cells, CAR T cells with an anti-CD19 cards were injected, and one contained a moiety, a co-stimulatory moiety called BB4 or the CD137. That's the other name of that. And the other cards, and that was a mix of those cells in that case, were CD19 CAR and then only the CD3 SETA moiety. So it was simply a first-generation CAR mixed with a second-generation CAR. The reason for that was that they had the plan to go into the clinic with both of them, and then they put it together in this animal study. And after discussion with FDA, while the study was running, they changed it, and they went only with this second-generation CAR into the clinic. But FDA still accepted this kind of assessment here from that. That's very interesting, and that was in 2009. Okay, so then you look, after certain days, you look on several parameters. What I've listed here is the amount of ALL cells in the periphery of the mice. So you see here that in the tumor control, which only received ALL cells, you see that a lot of cells are there. Then you have another control after 35 days where entransduced T cells were injected. They do not contain the CAR. And you also see that a lot of those ALL cells survive. Also, some of them were killed, and this is by the reactivity of the T cells due to their MHC-restricted killing of the cells. And so that's the active control, the inactive control then for the CAR T cells. Then what you see here are high dose, mid dose, and low dose of CAR T cells. And in the case of the low dose, you see also that it was not completely efficacious in those animals. If you go on now to day 49, you see again that also in the untransduced T cell control, you have more ALL cells, whereas the picture remains the same almost in the CAR-treated dosage groups. If you go longer than day 57, then you see that the tumor-only control is no longer existing because all the animals died of the tumor. But you have still the untransduced T cell control, which is here, with even more animals developing tumors. And you see also that in the low-dose cart, more animals now are present with the tumor here because the low dose was not efficacious. And we have an efficacious result here for the high and mid dose. Day 70, it's pretty the same picture here. So what does that tell us, and how does that relate into the effectiveness or into the suitability of such a study, for example, for dose selection in the clinic? So if you look at the cases of graft versus host disease in those animals, and that's something which is inherent of this model, because you put human cells into mouse in a mouse environment, then you see that with the low dose of the CAR T, you have less cells, of course, but you also have less graft versus host disease at the same time. And if you look at the high dose, then you see that with the high dose, I have to see, the orange one is the entrance-induced T cells. They produce a high number of GVHD deaths or premature sacrifices of animals. And if you look at the CTL19 cells, the positives, the CAR-positive cells, they also produce more GVHD-related dropouts, but less than the MOC cells. And this is because they have something to do with the tumor, and that may distract them from attacking the tissue of the mouse. And what you see in the mid dose here is that with the tumor, you have with the CAR-positive CTL19 cells, you have a low number of dropouts by GVHD, while you have the efficacy in this group, and only the MOC cells produce a higher number. And this shows that for those kind of studies, non-clinically, you have to establish an optimal balance between the tumor load, because animals should not die too early, and the appropriate amount of CAR T cells, because you don't want to produce them too many GVHD reactions, but you want to see efficacy in those studies. And nevertheless, you always have a confounding factor here, which is GVHD, which you have to balance out here. And that is one of the reasons why this is sometimes really difficult to use these kind of studies for establishing a first-in-man dose. And now we can mitigate that. I will come later to that. Okay, so value of animal models for preclinical safety assessment of the CAR cells. Pharmacological proof of efficacy can be demonstrated in these immunocompromised mice. However, as I said, we have limitations in predictive toxicology evaluation. And the prediction of a safe cell dose for first-in-man trials, I just explained that here. This is summarized again. What you have to do is to consider an optimal balance between the three criteria here, as shown on the slide before also, that you have a meaningful, at least one meaningful dose group, which is showing you the efficacy of your product and the proof of concept also in animals that it's working. And what is important to consideration of the optimal, okay, that's what I said. And currently a considerable clinical database with CAR T therapies. So we have a lot of CAR T therapies out, not only Kymriah, it's also Yescarta, it's many other companies. There are a lot of clinical trials going on. There is a lot of experience already out there. You can use that to find an efficacious cell dose, which enables you then for new CAR T products which are established which are established in the non-clinical behavior, and thus may support to define a first-in-man dose for the CAR T. So if you have a clinical database of a registered CAR T, you have all the safety data, because they need all to be put into the big databases, and they are followed also by health authorities on a regular basis. And you have animal models which supported in the beginning these first-in-man trials of this particular CAR product. Then you can use these animal models with these old CAR T and use the modified, either you have modified the manufacturing process or you have modified a small modification of the vector. Then you can compare that directly in the same animal model, and you see the differences between the old and the new. And this can help you to identify a factor by which you reduce the cell dose in comparison or in relation to the existing card. And this may lead you then to the new clinical trial and where you can apply that. So that could be one possibility, how you mitigate the limitations of those non-clinical studies. And I think we are not always starting from scratch. That has been done in 2009 by Carl June and closely followed by others. We are also relying on existing data where we have a lot of experience, and we need to have also at the same time always a good scientific rationale for doing that. Okay, prediction of toxicity, I said that already, is very limited. Biodistribution also taken with great caution because it may largely be driven by GVHD reactivity of the human cells in the mouse tissue. Yeah, just to, I have 10 minutes left, I can just explain a little bit on what was also found out in the recent years, how that works. Here you see a CAR T cell which is attacking a tumor cell by the CAR. At the same time, the T cell, of course, interacts with monocytes, macrophages, endothelial cells, and one possibility of this interaction is the CD40 ligand bridge, which can be formed between those cells. CD40 ligand is expressed on the T cells here. And with that interaction, the monocytes, macrophages may become activated. They release cytokines, for example, and other inflammatory markers. And here you see one of the most important for cytokine release syndrome, also in terms of clinical management, interleukin-6. But also interleukin-1 is released by those cells, and that's why in the follow-up of a cytokine release syndrome, in some cases, the neurotoxicity can be seen in the patients here. That's one of the links to that here. And it's indirect. It's not directly by the T cells. And CAR T cells, there had been papers out describing or suggesting that CD19 is expressed on peripheral cells, on the microvasculature, on neural cells in the brain, in the human brain. That turned out not to be true because they did an mRNA database analysis and they used mRNAs which were truncated, which appear in the brain, but fully functional mRNAs do not appear in the brain, and that's why CD19 is not expressed in the brain. So it's a true indirect pharmacodynamic exaggerated effect of the CAR T cell therapy. Okay, we come to that later. Okay, so what was investigated to somehow improve that situation? Here we have a possibility presented in 2018 at the European Gene and Cell Therapy meeting in Italy by the group of Attilio Bondanza. They used an immunocompromised mouse with 3 additional human genes, IL-3, GM-CSF, and stem cell factor, which makes it possible to establish a human blood system in the mouse. And with this model, they could, at the end, to be very short here, they could show that they had clinical signs of cytokine release syndrome, and they also could show that there were, for example, increase in interleukin-6 in those animals. So, however, the model depends on the use of human hematopoietic stem cells to establish the blood system in the mice, and that may not be suitable for routine testing. The other study was a homologous model in rhesus monkeys, also published in 2018. They developed a rhesus monkey-specific vector and treated the animals accordingly, and they were also able to show CRS effects and even neurotoxicity effects in those animals. And they could show also that CAR T were present in this cerebrospinal fluid after 1 week, and they infiltrated also the brain. However, this is an SIV-based vector which was used, and it's not a clinical product, so it's a homologous model, and the use in a routine toxicology assessment is really questionable here. Nevertheless, these kind of studies may be useful for doing scientific background work. So they have their value, but I think not for toxicology assessment. A standard toxicology assessment. So my summary here is target selection and validation is critical. Clarify potential cross-reactivity of the CARs, keywords on target, off tumor, off target, off tumor. CAR T cells are a fully human product. In case of autologous products, each patient receives a unique badge. Make sure that the non-clinical badge is representative for forthcoming clinical badges. Non-clinical safety testing is of a representative clinical badge faces limitations in animal models, as I explained. Vector safety, LISA on pre-infusion samples and from patient samples required per clinical protocol. I think that is, I forgot to mention here, that we have a long-term follow-up study for Kymriah, of course, also, up to 15 years by health authority requirement and guideline requirement. And here, in cases you have clonalities in the patients, in T cells, you are required to do a lentiviral insertion site analysis. But in our view, it is not required for same or comparable vectors, but different transgene. And we also have cases where we changed only the manufacturing procedure and had this, in principle, the same vector. And we also did not do LISA studies for that. And we were allowed to enter first-in-man clinical trials with that. Homologous animal models are possible, but the use of them is questionable, and they are also really not recommended by health authorities for supporting your clinical product directly. Homologous animal models, as I said, may be useful in mechanistic investigation of identified clinical adverse effects. Clinical hazard identification in animals is very limited. Thus, risk-based approach should be applied already in the pharmacological target validation phase. I think that is, the EMA has a very nice guidance about the risk-based approach out that is really explained, and especially for small biotech companies, it's really helpful to look at that. Not only if you want to, or if you want to not go for a clinical trial in Europe, but I think these are fundamental questions you can use and you can go through, and it's also useful for other countries or other regions of the world. Okay, so I'm not going into that. It's only to show you that the innovative activity in this field is immense. It's really huge. A lot is going on there, and I think the next decade of CAR-T development will be really, really exciting. And with that, I'm at the end. I thank you for your attention. So there were some questions. I don't know how to deal with that. Shall I answer them now, or how would you like to proceed? Okay, so there were some questions submitted before the talk, and the first was, what are the essential test items in a non-clinical trial of CAR-T treatment? I think I have gone through that, and I understand non-clinical trial more as non-clinical development, because the limitations of non-clinical pivotal animal studies, I think I have explained that. So I think it's the whole package. You have to go through target validation, pharmacological efficacy in an animal model, and vector architecture, if it's the same as the previous vector or another vector, or if you have a completely new vector, then I think it may get a bit more complicated. So that has to be considered then. During non-clinical trials, there are cases where additional trials are required due to the failure to meet the criteria when entering clinical approval. Please introduce failure cases to avoid the same mistake. I think I had one example from the literature that was this renal cell carcinoma trial where they tried to target with a card the carbonic anhydrase gene on renal carcinoma cells, and that went wrong. And this is something where you really have to look at your target distribution. The density of the distribution, is the density on tumor cells higher than in healthy cells, then the affinity of your card. You may want to play with your CAR in the affinity to reduce the binding to healthy cells in comparison to cancer cells. Also, I have to say that CAR cells are very effective. There was an investigation I heard from that less or almost 20 molecules on a cell are sufficient to initiate this interaction between a CAR cell and a tumor cell, and then the CAR cell is killing that. So it's very efficient. How do you set up criteria for non-clinical efficacy assessment dose for CAR T treatments? Okay, I think from my view, I have answered that. It's just because we are not starting from the scratch. We have already clinical data, we have non-clinical experiences, and we are not always changing the complete process or the complete vector. We are playing with that. And then you can follow this parallelogram approach where you go from the clinical experience, non-clinical, new non-clinical, and then you try to extrapolate that for a dose in the clinic with the safety factors you need for that. Okay. I'm happy to answer more questions if there are.
네, 필드에서 지금 질문 있으신 분 계실까요? 네. 저쪽에.
카티 비임상 그 사례에 대해서 소개해 주셔서 강의해 주셔서 감사합니다. 요즘에 그 알로제닉 카티에 대해서 많이 개발 중에 있는 걸로 알고 있습니다. 또한 그리고 노바티스에서도 그 알로제닉 카티에 대해서 개발이 되고 있을 걸로 생각을 하고 있습니다. 홀로 그 알로제닉 카티가 그 오토로고스 카티와 다르게 수행하고 있는 비임상이 있는지 궁금합니다.
I have to say I'm not aware if there's an allogeneic CAR development at Novartis. It must be very, very early then. But nevertheless, it's here you have to consider that if you use allogeneic T cells, you need to have also a match of those T cells with the recipient cells. And because you have an MHC of the donor cells and an MHC of the recipient, and something may happen that there is a reaction then at the end. In some cases, not Novartis is doing that, but other companies or universities, they're trying to knock out the MHC, for example, to avoid that reaction. And by that, they could improve that a little bit. And then, of course, here you have the same limitations also in animal models, because these are human cells you put into animals, and then the same limitations you have here. And then it's also that it's not really clear if knockout of the MHC is really sufficient to avoid all of these side reactions then.
안녕하세요. 식약처 이재린입니다. 네, 그 카티 세포 치료제 안전성 측면에서 그 온타겟 오프튜머 관련한 질문인데요. 지금 현재 제출되는 자료는 주로 휴먼 티슈 패널 휴먼 티슈 패널 어레이나 아니면 멤브레인 프로틴 어레이 같은 방법이 주가 되고 있는 것으로 알고 있는데요. 어떻게 보면은 민감성 측면에서 충분하지 않은 방법일 수가 있는데 노바티스에서는 이에 대한 다른 방법을 개발하고 있는 게 있으신지, 아니면 그 인실리코 데이터 분석이나 3D 컬처를 통한 방법에 대해서는 어떤 의견을 가지고 계신지 궁금합니다.
Okay. Yeah. First of all, it's the company with the microarray has improved that. They did some sort of tethering of the molecules, that they have a more distance to the membrane where they're attached to, and that may also avoid that some spatial conditions may inhibit the binding of the potential binding of a car. So that's there's an improvement ongoing here. And what Novartis is doing, first of all, I had one example, the suggested expression of CD19 in brain cells, in neural cells. Then we do special investigations of that with immunohistochemistry, with fluorescent in situ hybridization to follow that up. But this is all done when something is coming in a predictive manner. I think you can look at the epitope and see where the epitope is elsewhere presented. You have databases for doing that. And I think there are also methods out where you can consider of the conformation of this epitope, if it's visible for a car or if it's hidden for a car. I think there are database possibilities out there which you can use, and they are used.
혹시 네, 저쪽에 좀
예, 안녕하십니까. 저 식약처 홍영기라고 합니다. 그 발표 잘 들었고요. 그 렌티바이러스 삽입위치 분석 시험 관련해서 혹시 이제 가능하시다면은 그 이제 시험이 잘 수행이 됐는지를 이제 확인하는 작업을 이렇게 어떤 방법으로 수행하셨었는지, 뭐 물론 이제 현재는 그 NIBSC에서 제공하는 그 표준품이 이제 있는 걸로 알고 있기는 한데, 개발 당시에 혹시 이런 부분들을 어떻게 고려하셨는지 소개해 주시면 감사드리겠습니다.
So your question is pointing to the validity of the LISA. Okay, so as I said, I'm not an in-depth expert of this particular test, but they use positive controls. They, of course, have validated their assay, and in case of the gene work, it's now called Protagene because it was bought by another company. We did also a Novartis audit, and also they have certain certifications on how they do their work. So that's the formal thing. And this lab and also the Bushman lab were involved in the very first cases of identification of insertions into the LMR2 gene, which was happening with these so-called bubble boys with these severe combined immunodeficiencies. So they have a long-standing experience, and also they are very active in method development in this area here. And of course, they use appropriate controls to show that the particular assay was running in a way that it is valid. So that's very superficial what I can tell you to that.
네, 그러면 그 시간 관계상 추가 질문은 받지 못할 것 같습니다. 네, 지금까지 저희 이 카티셀의 비임상에 대해서 설명해 주신 우리 율리 박사님께 뜨거운 박수 부탁드립니다.
Thank you for inviting.